is, again, how many did we produce so far? We produced 4 ATPs through glycolysis. I'm repeating because I just through glycolysis and through the Krebs cycle. All right, we need to produce another 34 ATPs. We produce it through the electron transport chain. And we do all these NADHs and NADH2s, and where do they come from? They come from the Krebs cycle and the process of glycolysis, right? And where does this occur again on the mitochondria? The cristae, the folds of the inner membrane, the cristae. So that's what we're looking at here. Key things: we have all these proteins. They are protein pumps. Remember we talked about energy coupling, right? Remember we saw the proton pump from the last chapter, talking about how we can use ATP to pump protons, hydrogens out. Right, that's all we have here. The proteins on the electron transport chain, they are simply proton pumps. They're going to pump hydrogen ions. All right, they're not going to be powered by ATP though. They're going to be powered by the electrons, the oxidation reduction reactions that have been going on here. All right, so NADH, there's an H there, don't lose it. Right, NADH comes up. Is NADH being oxidized or reduced at the beginning of this process? NADH is going to have the electrons taken off, right? Oil rig, Leo Ger, right? Oxidation is losing electrons. NADH is losing the electrons to the electron transport chain. So NADH is being oxidized. It's giving up electrons, okay? So the electrons are passing on here. They're high energy electrons. As the electrons move through, they provide the energy to pump these hydrogen ions through, right? We're pumping hydrogen ions. We're pumping these protons from an area of low concentration to an area of high concentration. What are we creating? We used this word before. A concentration gradient, even more specific.
Electrochemical.
An electrochemical gradient. So we're creating this huge electrochemical gradient. Because hydrogen ions are charged, can they move through the membrane themselves? Can they move through the inner membrane themselves?
No.
No, they cannot, right? So we're creating an electrochemical gradient, right? That the only way for these hydrogen ions to reach equilibrium is to flow through the other protein involved here, which is the enzyme ATP synthase. What do you think ATP synthase synthesizes? ATP, very good. So we create this electrochemical gradient. The only way to reach equilibrium are for the hydrogen ions to power through the ATP synthase. It works very similar to hydroelectric power, right? Water flowing through a dam. All you have in hydroelectric power is you have this turbine underwater. When the water flows downhill, it spins the turbine, right? And it produces electricity. There's magnets and some wires, but the spinning of the turbine essentially produces electricity. We don't need to go into the details there. This ATP synthase, when the hydrogen ions move through, it literally starts to spin the ATP synthase. And it provides the energy to take the endergonic reaction, or to form the endergonic reaction of ADP and phosphate, or a phosphorus group, into ATP. This right there, that phosphate group, it's called PI, not as in like 3.14, but it just means inorganic phosphorus. It's always noted as inorganic phosphorus. So ADP plus the phosphate group, the energy from the ATP synthase, this enzyme here being powered by the hydrogen movement, powers the production of ATP. This whole process is known as oxidative phosphorylation, because oxidation reduction is powering it, right? NAD plus, FADH2 are being oxidized when the electrons are ripped off. What's ultimately being reduced in this process? What's the ultimate oxidative agent? Why do we call it an oxidative agent? Because something that's very good at being an oxidative agent is an atom known as oxygen. So what's the final electron acceptor here? What's the final oxidative agent? It is oxygen. You can't just keep pumping electrons on here, right? It'll clog up. Something needs to be the final electron acceptor to really draw and pull this whole process. It's not just a push here, it's also a pull on this side from oxygen, pulling on those electrons. That's where the energy comes from as well. So oxygen picks up the electrons coming through. It also picks up the hydrogen ions coming through. And it forms, at the end of this, high quality H2O, water. So oxygen is a final electron acceptor. It picks up the electrons, it picks up the water, and keeps the energy in the electron transport chain. Okay, so this whole process is oxidative phosphorylation. These proteins here are technically the electron transport chain. The ATP synthase is technically not a part of the electron transport chain. It's a separate component. It does a different function. But the other word that's up here that we should be familiar with is chemiosmosis. And that's simply, it's a big word for what we've already talked about. The flow of the hydrogen ions from the high to the low concentration, trying to get back to equilibrium through the ATP synthase, that flow of the hydrogen ions is known as chemiosmosis. So over here, this is not chemiosmosis. Chemiosmosis is just the flow back through the ATP synthase. And that's a word you should be familiar with. And that's it. For every one of these NADHs, we produce 3 ATPs. For every one of the FADH2s, because it comes in further down, right? We don't quite push as much hydrogen, it produces 2 ATPs. So if you remember back, we go back to our totals there. The question I just asked you, how many total NADHs have we formed? How many in glycolysis? 2. How many in the Krebs cycle? 4, but that's per pyruvate, so how many per glucose? 8. So we get 8 out of the Krebs cycle per glucose molecule, 2 pyruvates, and we get 2 out of the glycolysis, so we get a total of 10 of these through glycolysis and the Krebs cycle. How many FADH2s do we get? That's just in the Krebs cycle. We get 1, but that's per pyruvate, so per glucose we get 2. We get 3 ATPs per. We get 2 ATPs per. So these 34 are produced by phosphorylation, I can't fit it, and then plus the 4 we've already made through substrate level phosphorylation. There's your 38. Okay, so that's why oxygen is so important. Without oxygen, what happens to this electron transport chain? Shuts down. Because without oxygen, do we have oxygen to be the final electron acceptor here anymore? No, there's nothing there to pick up, to pull on the electrons. The energy in the system is gone. It's not the energy of NADH just giving up the electrons. It's the energy of the oxygen pulling the electrons through as well. So you lose oxygen, this system shuts down. If the electron transport chain shuts down, the Krebs cycle shuts down. Right? Therefore, the only process that can still occur is glycolysis. And we'll kind of deal with that now. So that's the big picture. There's our 38. Now, here's the wiggle room. Because you always hear 36 or 38, and here's a little bit of a wiggle room here. And it goes back to... It does. Glycolysis is actually goes on aerobic or anaerobically. Okay. So if we go back to this image right here, this is where the wiggle room is. Now remember, NADH makes more ATP than FADH2. This isn't a huge point, but it's just so you can understand the wiggle room. When your NADH is formed in glycolysis, it needs to pass through the membranes of the mitochondria to get to the electron transport chain. When it passes through the membrane, it actually goes through a process where it could form as NADH or FADH2. If it happens to reform as FADH2, right, this produces 3 ATPs per, this produces 2 ATPs per. If it happens to reform as FADH2, those two NADHs become FADH2s, which means that you're minus 2 ATPs, which means it takes you from your 38 to 36. So that's it. That's not a major thing, but that's just to explain the wiggle room, why we say 36, 30. It's not that we don't know. It's that it can reform on the other side of the membrane either as NADH or as FADH2. Everyone okay? All right. That's it. That's cellular respiration. But what happens if there's no oxygen? Right, glycolysis over, you go into fermentation. You don't die right away. You will eventually die, but you don't die right away. So here's kind of, and let's just kind of clean this up. I have a few more slides that show the same thing that I will show you. So here's the electron transport chain. Here's kind of the simple honored biology version of it. Just if you like this one better, it's just as good. It's just kind of a little bit less going on, but it shows you everything. Right, NADH, FADH2, hydrogen ions, ATP synthase. It shows that spins, ATP, right? And here is the reduction of oxygen to form water. So this and this are showing the same thing. This is showing kind of all of the different proteins that are involved in this process. There's ferredoxin, there's cytochrome A, cytochrome C, cytochrome B, right? There's all these different things. Don't memorize these words. The only thing you need to know is that in the electron transport chain, proteins can be referred to as cytochromes. That's what you want to underline. Underline the word cytochromes or write down the word cytochrome. So when you hear that word, you associate cytochromes with the electron transport chain, just in case that word comes up. I don't think I would ever quiz you on cytochrome, but in questions, the term cytochrome could pop up. And you just need to associate cytochromes with the ETC. All right. This right here is just showing you the ATP synthase. We'll watch it for fun. It's just giving you a little bit of how the action works. Oh.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
So again, this process of the flow of the hydrogen ions is known as chemiosmosis. So that's where the term comes in, the flow of the hydrogen ions through the ATP synthase.
The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport.
Okay. So that just kind of gives you a little bit of an idea. This is just talking about the hydrogen ion gradient. You can kind of read that on your own if you want, but we've already kind of mentioned it. Here's your totals, right? So here are the 2 ATP, 2 NADHs formed in glycolysis. Here's the 2 ATPs that are formed, and so forth. These are the energy investments. These are the product of ATPs. We get 2. And you kind of go down, you can kind of see the total production of 36 ATP. So it's just kind of looking at the totals. You can kind of, we've done that ourselves, so you can look at that again if you want. Here's another way we look at the totals, right? Here's what we produce through substrate level phosphorylation, right? Here's all the electron carries. You can see the either or situation, right? Here's your 32 or 34 ATPs here. So again, another slide just kind of highlighting totals there. Okay, but what happens if there's no oxygen present? Right? This is really what we're going to finish up on today. What happens is we move into fermentation. Fermentation is the process of producing energy from glucose in the absence of oxygen. So basically, what happens is the Krebs cycle and the ETC shut down. What can still function is glycolysis. Glycolysis still occurs. Glycolysis does not require oxygen. All right, so fermentation is anaerobic respiration. Glycolysis produces ATP. It is not nearly as efficient because we only get 2 ATPs per glucose compared to the 38 we were getting in the presence of oxygen per glucose. This is just showing kind of the breakdown. Right, glycolysis occurs. Pyruvate is produced. That happens in either aerobic or anaerobic. If oxygen is present, that pyruvate will enter the mitochondria. If oxygen is not present, that pyruvate will enter the second stage of fermentation, which is lactic acid fermentation or alcoholic fermentation. Here's kind of our breakdown. I'll give you a moment if you're taking notes here. So alcoholic fermentation, right, is releasing carbon dioxide and water. Yes. When you are allowing yeast to work, you're allowing your bread to rise. It's producing carbon dioxide. That's what's causing it to rise. The gases that are forming inside your bread. It's also producing alcohol. You don't become intoxicated when you eat bread, though, because when you cook it, the alcohol burns off. It's done. But yes, there is an alcohol content in your bread as it's rising, and that rising is caused by carbon dioxide gas being released. So that's alcoholic fermentation. The other type of fermentation is lactic acid. This is what we undergo, what animals undergo. And what happens is they both serve the exact same process, or the exact same point. The difference is here we produce carbon dioxide and ethyl alcohol. Here we just produce lactic acid. So you don't start to bloat and fill up with gases if you start working out too hard, right? You don't produce carbon dioxide in your muscles. At least in this process. And really, this is it. This is all it is. Fermentation is very simple. We do not get energy from the fermentation process. we get energy from glycolysis. But we have a problem. When we're going through aerobic respiration, right, all of those NADHs, all of those FADH2s, all of those electron carriers are being oxidized through the process. Therefore, they're being recycled. Right? Once NADH is oxidized back to NAD+, they can go back to the Krebs cycle, can go back to glycolysis to work through the process again. Right? Who might be laughing?
I looked at the picture and then I got it.
Oh, Jake and Kelvin?
Yeah.
So in aerobic respiration, right, we're regenerating our supplies of NAD+ plus and FADH2, right? The electron carriers we need for this process. With glycolysis, there's no regeneration of it. We're going to burn through all the NAD+ plus in your system, and then glycolysis is going to shut down. So we need to couple another reaction with it to make sure that we regenerate NAD+. plus. That's all fermentation does. It does not produce more energy. It simply allows glycolysis to occur. So with that said, here is glycolysis, the short version of glycolysis. Glucose, production of 2 ATPs, NAD plus is reduced to form NADH. In this process, we get pyruvate. In this case, we're looking alcoholic fermentation. That pyruvate is broken down into ethanol, ethyl alcohol. Carbon dioxide is released, and the NADH is regenerated back to NAD+, which allows glycolysis to continue to occur. So from the fermentation part, there's no energy produced. We're simply regenerating supplies of NAD+ to allow glycolysis to continue. The energy is produced from glycolysis. And again, they both serve the exact same purpose. This one is alcoholic fermentation, producing carbon dioxide and alcohol. This is lactic acid fermentation, simply producing lactate or lactic acid. Again, the only thing that's happening is we are regenerating NAD+. We far prefer aerobic versus anaerobic. So this is just kind of giving you a breakdown of it, right? Saying why, what both processes do, right? Saying that cellular respiration, aerobic respiration is far preferred, is far more efficient. And this is just talking about a few different organisms. We are aerobic, although we can undergo anaerobic respiration. Some organisms do not like oxygen at all. Right? They're known as anaerobic organisms. A lot of times they're bacteria. A lot of times they're in cuts. I've mentioned this before. Hydrogen peroxide is just H2O2. You can use it as a disinfectant. When you put it into a cut that's infected, the H2O2 breaks down into water and oxygen, and it floods the cut with oxygen, and it kills all that anaerobic bacteria, causing your infection. So we have some organisms that are obligate anaerobes. These are key vocab words to know. So obligate anaerobes are typically, for the most part, prokaryotes, bacteria, and they do not survive in the presence of oxygen. There are also facultative anaerobes. Yeast would be a good example of this. They can live in an environment with oxygen, or they can live in an environment that is in the absence of oxygen. So yeast, when you're creating beer, you're creating wine, you actually need to seal the unit and take the oxygen out, because it causes the yeast to shift from aerobic respiration to anaerobic, which is alcoholic fermentation, to produce the alcohol. But if you allow there to be a constant supply of oxygen to your yeast when you're fermenting, the process will occur and alcohol will not be produced. That's it, except for one last thing. We talk about this in terms of glucose. However, we don't just eat glucose every day. Right? We eat proteins, we eat lipids, we eat all these other different ones. Glucose is what we talked about, because glucose kind of takes you through the entire process. This is just looking at aerobic respiration, glycolysis, right, citric acid cycle, oxidative phosphorylation. But just kind of to understand, you don't need to know where they enter, but just be familiar with, hey, guess what? Proteins and fats can both enter these processes as well. They just enter at different points within the pathway. Right? Again, if you ever got a question asking about fats or proteins, where they enter it, there's going to be a diagram that goes along with it. You don't need to memorize this diagram where it is. Just know that they can enter the pathway as well.